Welcome to the nervous system part A. What I am and what I'm not going to cover. You have already memorized the parts of the brain. You've already memorized the spinal nerves, the cranial nerves. And to that end, what I'm going to do is just cover neuron to neuron transmission. And that has a lot to do with the fact that, in pharmacology, a lot of the drugs that are used to treat different nervous system pathologies, let's say like depression, is going to be aimed at e is going to be aimed at what's happening with the neurotransmitter at the synapse. And there are other drugs that are going to alter the electrical transmission. So since a lot of the drugs really are about that level of mode of action, I am not going to be covering like parts of the brain. Instead, it's going to be centered on how nerves talk to one another. Okay, so nervous system. It is a control mechanism. It is the quickest control mechanism in the body, and it operates by a feedback mechanism. The first is you have to have some sensory input. Environments can change within the body, without the body, outside of the body, and then uh, very often you will have some sort of sensory input about the change that has happened, because what we all want is homeostasis, right? We want things to remain the same. But if things change within and without, then you've got to do something. And so the sensory neurons are there to alert the central nervous system, the brain and spinal cord, which is integration, that something has caused a change in homeostasis. And of course, we want to get back to homeostasis. So the brain and spinal cord are going to analyze that information that's brought in by the sensory, and it's going to then respond to that with a motor output, or sometimes it's called effector with an E, effector output. And that output is to do something that would allow the body to respond to it in a way that would bring back homeostasis. Okay, so here is this individual wants to drink this glass of water. So the sensory input, I would not have used this as an example, but that's okay, we'll go with it. The sensory input, or the afferent pathway, meaning the first, is going to go to the brain and spinal cord. That's my integration. An interneuron means that it's a neuron that stays within the brain and spinal cord. Then the effector or motor neuron would then fire, and it would fire onto a skeletal muscle that would allow the hand to drink the glass of water. There we go. And that is the book's example. Now, you can organize the nervous system by structure and function. So if you're talking about structure, we have the central nervous system, which is the brain and spinal cord, and then all the neurons that either come into or out of the central nervous system is called the peripheral nervous system, because it goes to the periphery, like your arms and your legs. So, so there are paired spinal and cranial nerves that carry messages to and from the spinal cord and brain. All right, then we get into the whole issue of function. So you have voluntary control movement of skeletal muscles, that's called somatic. Everything else is autonomic. So when you think about how your cardiovascular system works, your digestive system works, renal works, etc., all of that is unconscious, is not under voluntary control. I don't have to think about, oh my God, I have to take a breath in now, and then I have to move a piece of food down my esophagus, and oh gee, I need to make some urine, by the by. that just happens automatically. So that is going to be your autonomic nervous system. It has two branches, the sympathetic and the parasympathetic. And again, that's not what I'm going to cover. It's something that you learned before, and it's fairly straightforward. In general, sympathetic tends to be about action, and parasympathetic tends to be about relaxation. But it's paired because a heart, for example, if it was increased in heart rate pathologically, you would want a sensor to go to the brain, which would then do a feedback with the effector that would slow down the heart to get you back to homeostasis. And that's that negative feedback that we talked about in the first chapter that we covered. Okay, histology. Let's say the brain, spinal cord is mainly made up of neurons, and they are specific for transmitting information from one to another, but there are some supporting cells. And I am going to give you an example on the next slide, and they're called neuroglia. And they're not neurons, they are supportive cells. What do they do? And there aren't a lot of them because the majority of the cells in the brain are neurons, but the minority are neuroglia. So it can provide a scaffolding for neurons. It can segregate and insulate neurons. That's what we're going to talk about. Or it could guide young neurons to proper connections during fetal development. And it can also promote health and growth of neurons in the brain and spinal cord. Now, the two that are about providing insulation, the myelin sheath, which we'll talk about in more detail in a few minutes, are the oligodendrocytes and their glial cells in the central nervous system, and Schwann cells, which are glial cells in the peripheral nervous system. More about them later. Okay, so these are oligodendrocytes. The blue are the myelin sheath, I think. Yes, they're definitely the myelin sheath. And to be discussed in a minute. And then this is the Schwann cell, where you can see the segments of a white fatty material that it is able to lay down. All right, so now we're going to get to the neuron, aka nerve cells. So they are composed of a body, axon, and dendrites. They are long-lived. They better be. They have a high metabolic rate, and their plasma membrane functions in electrical signaling and cell-to-cell signaling during development. A picture's worth 1,000 words. Here we go. The picture. So you have a lot of branching, both here. They don't show too much branching at the nerve ending, but there is. You can see some. And this tree-like branching means that it can communicate with a lot of neurons, because that's essentially the beautiful thing about the brain is you have millions and millions of neurons that branch, and they can talk and have very complicated conversations because they are so highly branched at the beginning and at the end. Your electrical stimulation goes from the dendrites down to the axon, which is kind of a straightaway, to the nerve ending, which is branched and releases neurotransmitter into that gap between neurons called the synapse. It does have a nucleus, and the nucleus is needed to make things like neurotransmitter. The axon is this straightaway. This particular one is showing you the myelin sheath, or pieces of fat material, because the myelin is made up of cholesterol fat. And I'll talk in a bit about how when you lay down this myelin sheath, this fatty layer, that you can conduct an impulse quicker. But some neurons have that myelin sheath and some don't. but this picture shows it with the myelin sheath. All right, I'm going to make quick work of this slide because it will be explained in more detail later on. But myelin, this cholesterol-based fat that can get laid down, it protects the axon. It's going to insulate parts of the neuron from electricity, and it will increase the speed of the nerve impulse transmission. All right, so how do we classify neurons? We can classify them as sensory, motor, or interneurons. So sensory neurons will take information to the central nervous system. Motor neurons will relay what the brain and spinal cord have decided to do, so they carry impulses away from the central nervous system. And interneurons are the neurons that are in the actual spinal cord and brain. Stay there. And stay there. There we go. And that's it. So part B is going to be more specific about the action potential and the neurotransmitter and synapse. See you soon.